I'm actually probably the only speaker today that's not going to have, I think, a single equation. So what I actually want to tell you about today are some of the things that we've been developing in my lab, motivated by all the exciting developments in ancient DNA, to think about what the world could look like if you had thousands and thousands of ancient sampled sequence. And as you can imagine, one of the huge limitations to enabling such a world is that today, ancient DNA sequencing is extraordinarily expensive. So if you sequence an ancient genome, about 1% of the reads on average tend to be human DNA, 99% of them tend to be bacterial DNA, and so it's just extraordinarily expensive. You have to have a Max Planck or be S.K. Willerslev and have the sort of backing of the Danish government in order to be able to do a lot of this kind of work. And so in order to sort of democratize ancient DNA sequencing, we've been working on techniques and methods to try to make it more affordable. That's also been in part motivated by a lot of the cool kind of evolutionary questions we might be able to answer. So I want to tell you about the technology we developed, and then some extensions of it to two problems. One is orthogonal validation. So one of the big problems I think we have in ancient DNA is that few samples have actually been sequenced more than once on two different technologies, and so we don't really have often a good understanding of what are true errors in the data versus real differences, except for some of the high coverage. And so if it's expensive to sequence a low coverage genome, you can understand it's extraordinarily expensive to sequence a high coverage sample. And then one actually cool thing that's come out of this is that you can think about the inverse problem, which is that imagine a sample where you have human as the contaminant, and you want to sequence the bacteria, and this is something like human blood, and we've actually just gotten some very preliminary results that say we may be able to do that, and so I wanted to share those and think about some of the analytical challenge that come down the pike in thinking about sort of metagenomics, where the metagenomic samples are actually at sort of low abundance. Okay, so I don't need to tell you about why ancient DNA is exciting or the problems that are involved. I do want to start with this story. So we got involved because of this character, this guy named Ötzi the Iceman. Ötzi was found about 20 years ago now on the border between Austria and Italy, and he's one of the most extraordinarily well-preserved ancient samples. So they found an entire sort of frozen mummy. So this is about ideal conditions. He's been kept on ice for 5,000 years, and they several years ago started a collaboration because when they found his genome, it turned out, when they found Ötzi, it turned out he'd not only sort of died in this crevasse, he actually had been murdered. And so I think the only way to solve a good murder is to sequence the genome. And so in a collaboration with ABI, they sequenced it. And even in this extraordinarily well-preserved case where you have a sort of bone biopsy, it's about 40% human DNA. And they did about an 8x genome, and we were involved in the analysis. For those of you who are interested, you can read the paper here. And one of the very cool results that came out of that analysis and what we were involved with, some of you know, we've been working on patterns of European diversity now for several years. This is from a paper we had with John Novembre. And when we PCA'd up Ötzi, so you would have expected him to sort of PCA up out here. So here's sort of our map of Europe. Here's the Iberian Peninsula. Here's Switzerland. Here's where you'd expect him to PCA up. But it turned out that good old Ötzi PCA'd up with these sort of 5 Italian samples that we'd always sort of swept under the rug. And so we sort of went back and go, yeah, what's up with those 5 Italians? It turned out they weren't just any random 5 Italians. They were Sardinians. And when we looked closer at the data, it turned out that Ötzi carried a mutation on his Y chromosome that had never been described before, and Peter Underhill, who's now a research associate in the lab, and of course, as many of you know, has worked for many years on the Y chromosome, genotyped that sample, or genotyped that marker in a huge collection of samples that he had. And one extraordinarily interesting result that came out of this is that the present day relatives, at least the male relatives of Ötzi, are all on the island of Corsica and Sardinia. Okay, so here's this 5,000-year-old dead guy, and we can tell you with sort of pinpoint accuracy at least about his male relatives. And so we became very interested in this problem, and you can imagine, right? There are sort of two scenarios. One scenario is that, let's see, really was Sardinian, and if you're Sardinian and you want to go on vacation, you go to the Alps, and maybe that's why he was killed. Who's this Sardinian guy here? And that's why he got iced, as it were. Or perhaps Europe 5,000 years ago looked very different than it does today. And really the only way to answer that is with lots of ancient samples from lots of different time points. And that's sort of where we decided to sort of take this. Okay, so now on to the technology. And this is really a project that's been led by Meredith Carpenter, who is a really extraordinary postdoc in the lab. Meredith was actually a PhD student here at Berkeley. And Jason Buenrostro, who's a PhD student at Stanford in Will Greenleaf's lab. And what we wanted to develop was a very inexpensive whole genome capture technique that would allow us to take that soup of molecules that have both human DNA and the bacterial DNA and whatever other things are in the soup and sort of pull out the human molecules. And we wanted to apply it to a real world set of samples, and so we got our hands on these 4 Iron Age and Bronze Age human teeth from Bulgaria, 7 different Peruvian mummies that we analyzed in collaboration with Eskay's lab, as well as a Bronze Age hair sample from Denmark. And then we wanted to intersect it with the sort of 1,000 genome data and ask, well, how well can we do with these sorts of data? Just taking, I mean, we didn't, certainly for the Bulgarian samples, we didn't know what we were going to get. We didn't know the quality of these. These were sort of representative of what you might expect to get from sort of samples that may be sitting around in people's museums. Okay, so the outline of the talk, I'll describe WISC in a bit of cursory detail for those of you interested. This has been published in a paper last year in the American Journal of Human Genetics. I'll then tell you about the orthogonal validation work, and then close with the work on metagenomics. And I also want to acknowledge Martin Sikora, who's a postdoc who's done much of the analysis that I'll be talking about, particularly for the second part. Okay, so here is how WISC works, and the details are really quite straightforward. So you can imagine that one big problem when you want to do capture is the last thing you want to do is introduce human DNA into an ancient DNA library, right? So you're not going to want to bait with human DNA. So one thing you could do is bait with RNA. And so the big problem then becomes, what is your source of RNA? And the real insight that Meredith had was, well, what if we were just to transcribe the entire human genome? So imagine taking human DNA, adding bacterial promoters, and just making boatloads and boatloads and boatloads of RNA. And that's basically what we do. So we can then ask, how well does the method work? But just think about it in that way. So you then go through and you add these probes to the RNA that you're going to use in your capture to pull them down. Okay? So the other part works in exactly the same way that ancient DNA normally works. So you take your ancient tooth, you do your DNA extraction. In our case, the red bits here are the human DNA. The black bits here are the environmental DNA. You make your sequencing library in the traditional way, and then you capture the library. And this is all in the paper. This is the way you would normally capture, and then you pull them out with the way you normally do capture using these streptavidin beads. Okay? That's all the molecular biology we're going to do today. So you can imagine that there's a ton of stuff under the hood here, and as anyone who knows me knows that I had nothing to do with the wet lab work. It was all Meredith, and so I'm happy to answer questions or honestly pass them on to Meredith and then pass on to you the answers. But now I want to tell you about the results. So here are the samples. So we had 4 iron Bronze Age teeth, and these sort of straddled from about 1500 to 500 BC. We had one nice hair sample, and that's this guy here. And then we had this mummy that perhaps looks like some of you felt maybe this morning if you went out drinking late last night. Or maybe how you felt when I told you I didn't have any equations in the slides today. Okay, and so here's some of the data. And what I'm showing you is the number of reads that map pre and post capture. So in blue here is pre-capture, and in red it looks like post-capture. And you can imagine this almost looks like an advertisement from the back of a magazine that you order reagents for, right? The red does much better than the blue, right? And so one thing you might ask is, well, how well are we doing, and are we biasing? And you really see, I mean, this is one of the most egregious examples of how well you do. You're kind of going from about 1 to 2% of the reads mapping to the human genome upwards to about 60% of the reads mapping to the human genome. But of course, we may only be getting a very small fraction of the human genome, right? So if the method's not working well, then there's not much to be excited about. So one of the things that we were concerned about is coverage, right? So if one thing you could do is say, map these to the human genome. Here are 2 examples. So here is one library, pre-capture, and these are all the sort of reads and where they're lining up to. And here it is post-capture. And what you're supposed to take away from this is that basically every place we're getting reads pre-capture, we're getting them post-capture. Does that make sense? And the likewise for another library, pre and post-capture. Okay? And you can imagine there are better ways of quantifying this in terms of overlap and so on. The other thing that became, and this is sort of just for one of the libraries, this is one of the Native American libraries that we ran twice. So this is for about 12 million reads that were sequenced. This is the post-capture, right, with about 200,000 reads. Post-capture, 50,000 of them are common to the pre-capture, and there were about 16,000 reads in the pre-capture that we didn't see in the post-capture. And you might imagine that if we sequenced more of the post-capture, we might get back some of these reads. Does that make sense? Okay. So here is some of the kind of methodsy bit that we became interested in, because one of the big questions is how to think about the problem in terms of efficiency. And Simone Gravel, who's a postdoc in the lab, has helped us think about this. In part, it's similar to a capture-recapture problem. So you can imagine that there's a trade-off in how much you invest in sequencing a given library versus what you're going to get out of it. Because the more you sequence the library, the more you're going to begin to sample the same molecule, right? You begin to get so-called duplicates. And so what I'm plotting here is the number of unique fragments that you're getting, and you have this so-called, right? I mean, not the law of diminishing returns, that as you sequence, you're beginning to get duplicates, okay? So what you'd love, and what we're basically getting with this technology, is that with the post-capture, so red is post-capture, blue is pre-capture, right? With the post-capture, we're basically already shooting ourselves into that world where many of the molecules are duplicates. Normally, if you have a lot of duplicates in your library, that's viewed as a bad thing, right? That means you have low complexity. But really, what's happening here is that there aren't that many unique molecules to sample in the first place. And so here's what you would get with the pre-capture if you were to be sequencing it out and out and out and out. You're kind of going to meet this same curve. And so the capture kind of allows you to get that curve. And then our enrichment is really the ratio of those two curves. Obviously, right, the more you sequence, the less meaningful the capture is, right? The more you sequence, the less there's going to be an enrichment relative to the two. And this is giving you a sense for the two. So this is the Danish hair sample. More enrichment here because there's sort of more duplicates. This is a more sort of complex library. And so there's sort of some less enrichment, but in general, right, you're still benefiting from doing the capture. Does that make sense? Everyone on board? Okay. So, and for those of you who are interested, Simone's also been thinking a lot about this issue of capture-recapture as it relates not only to molecules and sequencing of the DNA libraries, but also to identifying variants. I encourage you to talk to him. Okay. So, because of this issue of the duplicate reads, the number of fragments that are mapping to the human genome is really not a fair statistic to report as to how well the method works, right? Because really, if it's a kind of shitty library, right, then you're just going to be sequencing duplicates and not getting a lot of extra information. So one way that we thought about quantifying the amount of information that you're getting is as a function of a fixed number of sequencing reads. So this is something like per million reads that you sequence, how many SNPs are you finding pre and post capture. Okay, so here are the different libraries and the numbers here. These are all the sort of Native American libraries. P192 is one library that's particularly interesting. I'll go back to this. This is the one that we've now also sequenced on Ion Torrent. This is one of the Bulgarian libraries. And so you see that, even in this guy, this is a pretty good library. We're getting a twofold enrichment in doing the capture, so it's still quite beneficial to do the capture. Whereas for others, right, this one, you sequence 1 million reads, you're only getting 600 SNPs, but if you do the capture, you get 4,000 SNPs, right? So, there's quite a nice increase in the number of SNPs that you're getting, and we're basically getting usable data from every one of the samples that we tried. So this is actually, for us, quite encouraging, because it means that, in some sense, almost any ancient DNA sample is going to be amenable to some degree of analysis based on this kind of a capture technology, as long as, you have proper checks in there that you're doing a good job in terms of the uniformity of the sampling, which is really one of the motivations for why we then decided to look at this orthogonal technology question. Okay, so now on to what can you hope to learn, and does it really actually help us in any semblance of population genetic analysis? And so, one of the things you might imagine wanting to do is just a straight up PCA, intersecting the SNPs with sort of existing databases. So this is sort of the kind of collection of data you might use. And in this case, it's sort of 1,000 genomes, SNPs. This is the set of Asian diversity space. Here are the set of African samples. Here are the set of European samples. And the shockingly revolutionary conclusion here is that the Bulgarian ancient DNA sample clusters with the Europeans, which we've got nature and science sort of vying for that result right now. We'll let you know who goes for it. What's cool, though, is if you do the capture, you are able to begin to get into a world where you do begin to get some very nice potential subcontinental ancestry. And I'll dig into this topic a bit further, particularly talking about some of Martin's results. So in this particular case, the post-capture is giving us about 10-fold more SNPs. And so, now you're really talking and beginning to ask questions about, well, can we distinguish different, source populations? Is it possible to say who was more closely related to whom? And if you really sort of had blanketed and had a good set of samples, it may be possible. Okay, another example, this is our horrified Peruvian mummy who's just concerned about both the molecular biology that's being discussed and the statistics and just wants to have nothing to do with the talk at all. Perhaps they were also concerned that when we PCA'd them up, they PCA'd up with Asians and not necessarily with Native Americans. And this was a concern for the mummy because this mummy actually has blonde hair. And there was a sort of question about whether this mummy could be Native American or in fact could have some degree of European ancestry. It's actually a pre-Columbian mummy. However, when you go to the post-capture, you see that in fact it is PCAing up right where you expect of a nice sort of Native American sample. And so we're very excited to be following this up in some more detail. Okay, so to sort of summarize the sort of results for where we are, or how the method is working, for the positive capture, on average, you start with about 1.2% human DNA. Post-capture, we've gotten up to about 60% human DNA. We're working on improving this. Enrichment of molecules between 5 to 140x. Enrichment of SNPs from about 2 to 20x. And you're getting somewhere on the order of about 50,000 SNPs per million samples. Okay? So we really are quite excited about this and are now trying this on dozens and dozens of samples and exporting it to other labs. Yeah, David?
One question I have is I'm very interested in this. I think it's very exciting potentially. So one thing you'd like to do is get whole genome level of coverage with a large proportion of the genome captured. Yeah. So what is the largest amount of data from a single sample that comes through this process?
So I'll show you. It all depends on the endogenous DNA content.
Right. So I guess what is the largest complexity of a library that's come out?
I'll show you the data that we have on one library that we sequenced on Ion Torrent, which is the most complex that we have. That's coming up. In fact, thank you for the sort of lead-in. So one of the things that we're very interested in is this sort of this question of orthogonal validation and trying to see how well the data they're coming in and out are. So we've sequenced this sample in collaboration with ION, as well as on the HiSeq, as well, and that's supposed to be WISC, as well as using our WISC technology. And this is sort of a plug for where this began. This is part of this project that Martin is leading, where he has taken these set of published genomes, as well as our Bulgarian sample, where, and this we've now sort of increased this in the data that I'm about to show you. And I'll motivate it by saying if you take, and this is sort of the kind of going back to this question about where sort of Sardinia fits in relative to the rest of Europe. So Martin took these hunter-gatherer samples. So there's a Swedish hunter-gatherer data set that had come out from Pontus and a Swedish group. And they had a hunter-gatherer and a farmer. There's the Iberian hunter-gatherer. We put in Iceman and the Bulgarians. And so one of the main results of that analysis, and so if you use David's d-test and you look at Iceman, for example, so the d-test, as you'll recall, is a test for gene flow where you'd have, for example, here's Sardinia, here is our ancient sample, here's what we're comparing to. And so if you have, in this particular case, Iceman, this is sort of the z-score, and it's telling you, well, this is the d-statistic, and then this is sort of the level of the z-score. There's strong evidence for more affinity between the Sardinian and the ancient genome than any one of these. So evidence for, and in particular, the Finns cluster particularly out, and there's a reason for that separately. But so the take home is that the Iceman is closer to Sardinians than to Brits, CEU, Iberians, or Tuscans, and you can say that quite confidently. Does that make sense? And that's because of the strength of the, not so much because of the strength of the effect, but because of the strength of the data, right? There's a tremendous amount of data because this Iceman was sequenced to high coverage. So when you look at the other samples, which is sort of this motivation to taking it to higher coverage, so the Swedish farmer shows the same pattern as Iceman, so closer, the more clustering with the Sardinians of the farmers, be they the Swedish farmers, be it Iceman or our Bulgarian, than the hunter-gatherers, and the hunter-gatherers trend towards where the samples were taken. So both the Swedish and the Iberian hunter-gatherers are sort of closer to the Finns, Brits, and CEU. Okay? And there's lots of folks who are thinking about this problem and the impact that this has on models of for the peopling of Europe, and if we've got time at the end, I'll show you Martin's thoughts on that. But I just want to sort of focus in on this issue, which is this is all being driven, of course, not so much by the effect size, but the fact that the amount of data that you have, these confidence intervals are straddling zero. So if you could, increase the amount of data, you would do a much better job in being able to estimate the actual value of those d-statistics. Okay, so here's the data, or at least, let's begin to talk about the data. So we sequenced that Bulgarian ancient DNA library. In this case, this is what the Illumina pre-capture was. It's sort of about 13 million reads. From those 13 million reads, you call about 43,000 SNPs, and you have about 11 megabases of the human genome that this is mapping to. Okay, so definitively not a high-coverage genome, right? Our post-capture data, in fact, is even less. It's less than what we had in the pre-capture. That's why we call somewhat fewer SNPs in the post-capture. It's actually got less overlap, but the proton data was sort of through the roof. And here we've got about 467 megabases of sequence. And again, this is beginning to tap out. We've got, this is and I don't remember the exact numbers, but it's in the 20 to some percent duplicates, and we call about 400,000 SNPs. Okay, so now to get to this point, David, which I think is an important point. Here is perhaps a worrying slide, but I want us to think about this because I think it's an important slide. Okay? Here is the intersection of the data. So here's where all of the proton data is mapping. Here is the Illumina pre-capture, and here's the Illumina post-capture. Okay? So now let's think about what may be happening in this experiment. So we have two DNA extracts. Well, sorry, we have the same DNA extract. We've made libraries twice. One for the pre-capture and one for the Illumina libraries and one for the proton libraries. Now, when you sequence the proton libraries, right, you don't yet have a 30x genome, right? So this is mapping to 0.1x of the genome, right? And these guys are mapping to some other part of the genome. Luckily, I mean, we have good overlap between the pre and post capture. The reason the post capture doesn't cover everything in the pre capture is because we haven't sequenced it to higher coverage. We could sequence to higher coverage. Does that make sense? The other possibility, and there is another possibility, is that in both the library construction or in the capture, you're losing so many molecules that the whole thing is a concern for reproducibility. Yeah.
Do you know how this overlap looks as a function of frequency?
I don't, but I have some other results that sort of speak to this issue. I think it's largely driven by the coverage issue. If you could let me get that. Yeah.
I mean, we're doing capture, not whole genome capture in our lab, but capture of a set of SNPs.
Uh-huh.
And we lose 2/3 the complexity in the capture right now.
Yeah.
And so we got to. I mean, it's actually the data's usable, but it's a quite serious loss of complexity. So it's great not to be able to. I mean, it's very exciting in principle not to be able to reach our complexity through the capture.
I agree.
Yeah.
So what we need are more library sequence to higher coverage in order to address that issue.
Can you address it just as a technical, is this a sort of statistical question? I'm trying to think, do you actually need the higher coverage to?
So I'll take you to one place.
Can you compute statistics which don't need to saturate the library? In order to
I'll take you to one place where we actually do have high coverage, where we might be able to address some of these issues.
But I guess the question is, aren't there? I was just trying to think about this for our own work.
Uh-huh.
Aren't there statistics like do you not need high coverage to make this assessment? Like, can't you actually project or?
So you could take the curves, right, and those curves you could get, what is the expected overlap given the degree of coverage.
Yeah.
But I'll take you to one. Let me get to this point, which is that we do have some parts of the genome that are sequenced to quite high coverage, namely the mito, right? Because there are many, many more copies of the mito in the data than there are of the nuclear genome, right? So you've got 2 molecules of the nuclear genome in every cell, but you've got many, many more copies of the mitochondrial DNA. So if you look at the mitochondrial coverage, in the Illumina pre-capture, we've got something like, whatever you can sort of read it off on there, it looks about 1 to 2x, and we've got about 2 to 3x coverage in the post-capture, and we've got 20 to 30x coverage in the proton data. So from the proton data, we actually have quite good coverage and can make quite good reliable calls on the mito, and then ask, relative to the Illumina, how well are we doing? And this is some of the data and this is sort of literally just coming in, and I want to thank Martin and Meredith. And I actually view this as quite, potentially positive, right? Namely that if you look at where we're able to map pre and post capture and with the proton data, we're getting quite decent coverage in all 3 data sets in terms of the actual coverage of the mito from the whole genome capture, right? And the whole genome capture also has the mito as part of the capture, right? Because you're just making it from whole human genome DNA. Does that make sense? Now, just because we've got somewhat higher coverage of this, and this is actually an important point, the data is still sort of 1 to 2x data. And remember that the Illumina technology still has 1 to 2% error. So the actual quality of the data is not spectacular. So here's some of the mito coverage data. Here, and I'll show you several plots like this. This is post-capture, pre-capture, and ion, right? And you can see what the difference is, pre and post-capture. Right, and you're not losing, in terms of the number of molecules we had in the pre-capture and the post-capture library, we're basically getting everything we had, at least in our hands. And part of it is, we're not targeting specific SNPs, right? We're targeting whatever molecules are in the soup that are binding to the human genome. All right. Now, what do the, it could also be we got a ton of contamination in the data, right? So, maybe we're just, sequencing Meredith or something like that. Turns out that's not the case. The substitution types are exactly what you would expect from ancient DNA, largely C to T and G to A substitutions. They also follow the kind of characteristic patterns. And this is actually, so here's a kind of cool result. So you've probably all seen these sorts of results where the error rates go up towards the end of the molecule. But one cool bit is that there is somewhat of a difference between the proton and the Illumina patterns. And this is perhaps attributable to the way that these two libraries are made due to the AT overhang at the second position. Okay? So for those of you who kind of want to geek out on that, I'll leave you to think about that. Okay. Now here is actually some of the things we're now burrowing into. So if you now think about the pre- and post-capture library and what you've run, this is for the MITO in particular, because it's still low coverage genome sequencing, there's not a spectacular concordance. So there's about 24 variants that get called. 12 of those get called as non-reference in both. And then each data set calls 6 variants that the other data set doesn't, pre and post capture, at least running it through GATK. Okay? And this is due to the fact that you often have, 1, maybe 2 reads that are going to support a variant. Okay? And this is not surprise, folks, because when you have 1% error, right, this is going to happen. Right? So, yeah.
We've been experimenting with recalibrating.
Yep.
Particularly using the mitochondria.
Yes.
So when you say GATK, it may be that there's some clever pre-processing that will save you at least a little.
I'm sure there are. I'm sure there are. And I'll show you some additional results there that kind of speak to this. Yeah.
The mitochondria, there are a lot of chimeric inserts in the autosome, right? So when you go to call variants, it seems like you could have an issue with things mapping that actually came from elsewhere.
Potentially also an issue. Those are all very good questions. I'm sort of just giving you where we are with this. Okay?
The error remain in sequencing or PCR?
I think it's actually in the sequencing. And in fact, the error is actually probably in GATK. And I'll show you this sort of result, which is that we do have the proton data. So in the proton data, there were 6 sites that had coverage on the Illumina. Okay? And those 6 sites got called with high got called reliably on the post-capture. 4 sites got called on the pre-capture, and 2 sites were failed to be called by GATK in the pre-capture. I'll show you the data for those 2 sites, but the upshot is that in fact the reads were going in the right direction. So in fact the evidence is there. It's an issue of sort of calibrating and getting it to work. Perhaps the more worrying issue is there are about 20 sites that got called in the Illumina data that ION did not call, and we don't know if that's currently ION and that, I'm showing you the data, warts and all. This is literally data that we've been working on up until today that I was excited about and wanted to tell you. So these could be ION undercalls, or they could be Illumina errors. ION, because they've had issues before, tend to be very, very aggressive in their calling. Okay? But the point is, the only way that we can begin to ground truth this data is by beginning to get orthogonal validation. And there are almost no data sets that I know of where you have orthogonal validation for ancient DNA samples. And that's why we're trying to do this. Okay, so here are 2 examples where the reads, so here's the ION data, here's the pre-capture data that doesn't get called by GATK, here's the post-capture data that does call it, and it turns out the only difference between these 2 is the quality score that gets assigned by GATK in this pre-capture instance, right? So this is a false negative. It should have gotten called, but it just didn't get called in this particular instance. And the other one is the same scenario. Okay, one thing you can do after you sort of begin to deal with all these issues is make calls, and one thing that's very cool about this particular sample, it turns out to be a U3B mitochondria, and what's exciting about that distribution, first of all, if you sort of look it up on 23andMe. First population example is Bulgarian, so that's good. That's where the sample comes from, and it's also sort of the sort of Middle Eastern population center of diversity. And so one sort of model could be that this 3,000-year-old Bulgarian sample is part of that sort of initial Neolithic expansion into Europe. And there are a lot of these used that you actually tend to see in ancient DNA. We can also call the Y chromosome, and in this particular case, what's cool is that this is haplogroup E, Y chromosome, and as you know, this is kind of a classic group from the Bantu expansion, but you also see it in North Africa and some Southern European populations. And so, the sample assignments of the Mito and the Y make sense, and we're excited and beginning to take this to higher coverage. And again, sort of the bigger picture idea is that it's the beginning to open up more and more samples that could be amenable to this kind of dissection. Now on to some PCA, and I'd like to call this perhaps the worst PCA we've ever produced in the lab. No sort of offense. This is the pre-capture PCA for the Bulgarian sample. Of course, it looks like a Rorschach blob. You can read whatever you want. Well, I mean, in general, PCAs are Rorschach blots, but this is at least a Rorschach blot with more resolution, in which this is our sort of map of Europe PCA, which at least from the proton data, we're beginning to see that our Bulgarian sample is, by the incredibly sophisticated method of drawing this circle, is PCAing up near our 4 or 5 Sardinian samples. So that's where we are on that orthogonal validation project. We're quite excited about it and beginning to think about additional sources of sequencing that we can bring to bear to ground truth some of these samples. And again, if there are high coverage samples that we could try our capture on, we'd be happy to try them out and begin to build this sort of reference set out there that could be used. Okay, how am I on time, Andy?
You have 3 minutes.
I have 3 minutes. Okay. I'm going to just turn to the last bit, which won't take very long. So you can imagine that in the same way that we can do positive capture, instead of sequencing what you've captured, you could do some clever things and sequence what you didn't capture. So keep the unbound DNA. And you might ask, well, why on earth would you want to do that? And there are certain scenarios, particularly in metagenomic applications, where there's a lot of interest in low-level bacteria or viruses that may be circulating. I was just at AGBT last week, and Joe DeRisi gave just an amazingly beautiful talk about a kid that was seen at UCSF that had severe combined immunodeficiency syndrome, and the kid had been sick with an infection for a year and was just getting worse and worse and worse and worse. They finally sequenced a spinal tap. Turned out the kid had a form of meningitis that with all the other assays they weren't able to get it, and they did it by sequencing. From the I don't know how many tens of millions of reads, only 400 were to the spirochete. So you could imagine if there was some way to reduce the human, you might be able to detect it with far fewer reads. Okay, so we've done some mock experiments in this regard. This is work, not mock experiments, they're real experiments, but they're sort of samples that were set up. One is a saliva sample, and these were kind of designed for sort of a MiSeq, so about 10 million reads. In the initial library, about 50% of the reads map to the human genome, 50% sort of microbial. We do our depletion, and this is, to be honest, the best example we have. In this particular case, we go from about 50% human to about 3% human. And one way of quantifying this might be as an odds ratio, and that odds ratio is about 31.
Sorry, Carlos, what do you mean by depletion?
So
you actually suck out the human material from the sample?
Yeah.
So
you capture the human,
capture it like you use
you use whisk, and then you sequence the other bit. And here's some additional samples. So here's a cheek swab. And then here are 2 human soil samples. These are actually, does anyone know what human soils these are? These are the ones from the British. These aren't, the picture is of the footprints in Britain that came out from it. These aren't the ones we sequenced. Those would be very cool to sequence, but you know, we didn't get our hands on that. Yeah.
You just use WISC in the sense that you sequence or you make an RNA library for the virus and just do an analogous thing, or WISC is just...
That would be great if you know what virus you were looking for.
Right.
In our case, we don't know what virus we're looking for, right? And that's one of the main applications in metagenomics. So of course, one potential problem with this, right, that immediately comes up is you could very well lose the cool viruses you're looking for in your capture, right? That's one of the potential big concerns. Oh, so here are the, again, the depletions from 40 to 20%, 27 to 18%. So here, you know, again, very fresh results, sort of very much work in progress. You know, this is an odds ratio of 31. These are sort of more GWAS-like odds ratios, about 2 to 3. But you know, odds ratios of 2 to 3 are sometimes also very important, and we're in the process of trying to get those we're sequencing under the peaks, as it were, trying to get them to improve. And again, you know, very first kind of preliminary results on this. One thing that's a very kind of logical thing to try to do is ask, well, what's the frequency of the bugs pre and post capture, and you know, for lack of a better reason, just to sort of spread out so we could look at this in a bigger range, I plotted here the sort of square root of the frequency pre-depletion and post-depletion. So first, the bugs make sense for what you'd find in somebody's saliva, a bunch of streptococcus samples, you know, so what's happening in this particular case, these are things that, you know, we had some frequency of pre-depletion, and then post-depletion, we kind of lost. So, you know, in this particular experiment, we did lose some, but there were others that, you know, did pretty well. And the correlation here is about, you know, 92%, which is, you know, for a first-time stab at it, not a bad correlation. We're, you know, continuing to follow this through. Here's a second example. This is one that was a sort of cheek swab. This one's doing somewhat better. And in this particular case, this is actually a bacteria that causes gingivitis. I'm sure there are much cheaper ways of potentially diagnosing somebody with gingivitis, but in any way, this is what we got. So we're excited about this. We're following it up, and in particular, trying to get real samples where you have, you know, low amounts of the bacterial DNA that you'd like to sequence. And so, you know, could imagine transplant patients and other scenarios where this kind of sequencing might be useful. So with that, just want to close and say that, you know, we're still very much in the early days of getting this working. We've developed this gold standard set of samples, or we're thinking about trying to get these gold standard. And it would be good to sort of get a community to kind of rally around a set of samples that get sequenced using multiple technologies, so we begin to really ground truth these, and in the way that, say, Genome in a Bottle is doing this for some of the HapMap samples and some of the PGP samples. The depletion results so far look encouraging. We're working on reproducing them. We've had other experiments that so far haven't been as exciting, but we're working on it. It's coming down the pike. And the other cool thing is that you can do WISC for anything for which you have a DNA library. So you actually don't need a genome. So one project we have is a sort of dodo genome sequencing project where we're making the DNA baits from Nicobar pigeon. And, you know, we'll then build a Nicobar genome, but you don't need a Nicobar genome and, you know, sort of and a good Nicobar assembly, but rather just a source of the Nicobar DNA in order to bait. So with that, I want to thank the folks that have been involved, particularly Meredith and Martin. This is really a lot of their work. Will, my colleague at Stanford, who we've done a lot of this work with. And for any questions, I encourage you to ask Meredith and Martin. I'm happy to pass them along. Thank you.
So we're slightly into the coffee break, but let's take a couple of questions and see how that goes.
Yeah. It seemed like the depletion worked a lot better for saliva than for cheek swab and for human soil, based on this graph. Do you have any idea why?
That's n equals 1. So I wouldn't, you know. We don't know. We'll try it again. I wouldn't make a lot of it just yet. So the DNA content's very similar, so at least in terms of proportion. But you know, there are other things. You know, there are other approaches out there. So like NEB has a kit that pulls out methylated DNA, or kind of destroys methylated DNA, and bacteria don't tend to have methylated DNA, but this is what we're trying. Yeah?
So for sequencing like a specific pathogen population, people typically use a primer set that's specific to that pathogen.
Yeah.
You have to do this PCR step where your primer is seeking out this rare pathogen to see in human DNA.
Yeah.
It'd be interesting to if you could use WISP to take out that human DNA and see if you can increase the quality of your primer.
Yeah, I mean, normally you do this like 16S sequencing. If what you care about is ID, you would normally do the 16S sequencing. In this case, I think the reason to do this instead of 16S sequencing is that this would allow you to also get at whether you might get lucky if the pathogen had some kind of pathogenicity island or something. You could potentially find it here by sequencing to high coverage and building the genome that you couldn't necessarily do from doing the 16S sequencing, if that makes sense. Right, because the 16S is only going to kind of tag it.
Seems to me an awful lot comes down to the initial library complexity.
Yeah,
because
yeah, for sure.
If you're interested in the microbiome, microbiomes typically have a few abundant bugs and this long tail of extremely rare things. Anything that you do that restricts complexity, you're going to lose that tail.
Yeah,
So
yeah.
if you're interested in rare stuff, you're in trouble.
Yeah, yeah. So the other thing you could do is you could do the capture. So we had made with. So you could also do the capture pre-library, right? You can capture before you do the library construction, right?
Well, there is the issue of the complexity of the original sample, too.
That's also right. But no. So in our case, the reason we do the capture post is because the libraries are made in ancient DNA labs. So for the metagenomics.
Well, there must be a PCR. You've amplified it before you do the WISP, right?
Right.
All right, let's take a coffee break and be back there promptly at 11.
Thank you.